Dear viewers, Namaskar. Welcome to the lecture series on food technology. Today, we shall discuss about the topic microorganisms in foods. Under the subheadings 1. Introduction, 2. Primary sources of microorganism found in foods, 3. Common foodborne bacteria, 4. Common genera of foodborne molds, 5. Common genera of foodborne yeast, and at last 6th common viruses on foods. Introduction. Food serves as a growth medium for different kinds of microorganisms. Microorganisms enter into food and grow as contaminants or intended additions. Growth of microorganisms in food may spoil food quality or improve depending on the types of microorganisms and changes they bring about. Food are assessed for their quality in terms of physical, chemical, sensory and microbiological characteristics. Microbiological characteristics are assessed in terms of the microorganisms present in food, their characters, ability to change the quality, their influence on health of consumer. It is necessary for food microbiologists to become acquainted with the microorganisms important in food, at least to the extent that will enable them to identify the main types with their characteristics. Important microorganisms associated with food are bacteria, molds, yeast and some viruses. Knowledge of these characteristics and primary identification methods is necessary for the people working with food microbiology. Let us discuss now primary sources of microorganisms found in foods. The first one is soil and water. These two environments are placed together because many of the bacteria and fungi that inhabit both have a lot in common. Soil organisms may enter the atmosphere by the action of wind and later enter the water bodies when it rains. They also enter water when rainwater flows over soil into bodies of water. The second one, plants and plant products. It may be assumed that many or most soil and water organisms contaminate plants. However, only a relatively small number find the plant environment suitable to their overall well-being. Those that persist on plant products do so by virtue of a capacity to adhere to plant surfaces, so that they are not easily washed away and because they are able to obtain their nutritional requirements. Food utensils. When vegetables are harvested in containers and utensils, one would expect to find some or all of the surface organism on the products to contaminate contact surfaces. As more and more vegetables are placed in same containers, the normalization of the microbiota would be expected to occur. In a similar way, the cutting block in a meat market along with cutting knives and grinders are contaminated from initial samples, and this process leads to a buildup of organism, thus ensuring a fairly constant level of contamination of meat-borne organisms. Gastrointestinal tract. The intestinal biota consists of many organisms that do not persist as long as in waters as do others, and notable among these are pathogens such as Salmonella. Enterobacteriaceae members may be expected in fecal waste along with intestinal pathogens. Food handlers. The microbiota on the hands and outer garments of handlers generally reflect the environment and habits of individuals, and the organisms in question may be those from soil, water, dust and other environmental sources. Additional important sources are those that are common in nasal cavities, the mouth and on the skin, and those from the gastrointestinal tract that may enter foods through poor personal hygiene practices. Animal feeds. There is a source of salmonella to poultry and other farm animals. In the case of some silays, it is a known source of Listeria monocytogenes, a dairy and meat animals. The organisms in dry animal feed are spread throughout the animal environment and may be expected to occur on animal hides. Then animal hides. In the case of milk cows, the types of organisms found in raw milk can be reflection of the biota of the udder when proper procedures are not followed in milking and of the general environment of such animals. From both the udder and the hide, organisms can contaminate the general environment, milk containers and the hands of handlers. Air and dust. Although most of the organisms found in air and dust in a food processing operation, the ones that can persist include most of the gram-positive organisms. Among fungi, the number of molds may be expected to occur in air and dust along with some yeast. In general, the types of organisms in air and dust would be those that are constantly reseeded to the environment. Bacteria are unicellular microorganisms with small cell size ranging from 0.5 to 1 micrometer width and 1 to 5 micrometer length. Several bacteria are spherical in shape called cocci with a diameter of 0.5 to 1 micrometer. Several other bacteria are with different morphological structures like oval, rod, comma, spiral and spring-like shapes. Bacteria grow on wide range of nutrients starting from simple elemental nutrients to complex animal fluids like fetal cough serum. Majority bacteria are useful to human welfare and some are harmful, causing different infectious diseases. There are also important food contaminants and agents of foodborne diseases and food poisoning. It is necessary to know about these organisms. Some of the common bacteria are as follows. Acinetobacter. Gram-negative rats show some affinity to the family Neissereaceae. They are strict aerobes that do not reduce nitrates. Although rod-shaped cells are formed in young clusters, old cultures contain many coccoid-shaped cells. They are widely distributed in soil and water and may be found on many foods, especially refrigerated fresh products. Aeromonas. These are typically aquatic gram-negative rats, formerly in the family Vibrionaceae, but now in the family Aeromonidaceae. As the generic name suggests, they produce copious quantities of gas from the fermented sugars. They are normal inhabitants of the intestines of fish and some are fish pathogens. Alkali genes. Although gram-negative, these organisms sometimes stain gram-positive. They are rods that do not, as the generic name suggests, ferment sugars but instead produce alkaline reactions, especially in litmus milk. Non-pigmented, they are widely distributed in nature in decomposing matter of all types. Raw milk, poultry products and fecal matters are common sources. Acrobacter. They are gram-negative curved or S-shaped rods that are quite similar to the Campylobacters, except they can grow at 15 degrees Celsius and are aerotolerant. They are found in poultry, raw milk, shellfish and water and in cattle and swine products. Bacillus. These are gram-positive spore-forming rats that are aerobes in contrast to the Clostridia, which are anaerobes. Although most are mesophilic, cyclotrophs and thermophiles exist. The genus contains only two pathogens, Bacillus anthracis, which is known to cause anthrax, and Bacillus cereus. Although most strains of the latter are non-pathogens, some cause foodborne gastroenteritis. Burkholderia. Gram-negative rats that occur on plants, especially certain flowers, in raw milk and cause vegetable spoilage. They are significant pathogens in cystic fibrosis patients. They were formerly classified in the genus Pseudomonas. Campylobacter. These gram-negative spirally curved rods were formerly classified as Vibrios. They are microaerophilic to anaerobic. Corynebacterium. This genus of gram-positive catalase-negative rods were formed to accommodate some organisms previously classified as lactobacilli. They are heterofermentative and most grow at 0 degrees Celsius and none at 45 degrees Celsius. Gas is produced from glucose by some species. They are found on vacuum packaged meats and related products as well as on fish and poultry meats. Clostridium. These anaerobic spore forming rods are widely distributed in nature, as are their aerobic counterparts, the bacilli. The genus contains many species, some of which cause spoilage in humans, that is Clostridium perfringens causes food poisoning and botulism. Mesotrophic, psychotrophic and thermophilic species exist. They are important in the thermal canning of foods. Corynebacterium. This is one of the true coroniform genera of gram-positive rod-shaped bacteria that are sometimes involved in the spoilage of vegetable and meat products. Escherichia. This is clearly the most widely studied genus of all bacteria. Some strains known to cause foodborne gastroenteritis. Flavobacterium. These gram-negative rats are characterized by their production of yellow to red pigments on agar and by their association with plants. Some are mesotrophs and others are trichotrophs, where they participate in the spoilage of refrigerated meats and vegetables. Aphnia. These gram-negative enteric rods are important in the spoilage of refrigerated meat and vegetable products. Lactobacillus, gram-positive catalase-negative rods that often occur in long chains. Although those in foods are typically microaerophilic, many true anaerobic strains exist, especially in the colon and the rumen. They typically occur on most, if not all, vegetables, along with some of the other lactic acid bacteria. Their occurrence in dairy products is common. Many fermented products are produced and are also common on refrigerator stored vacuum packaged meats. Listeria. This genus of 6 species of gram-positive non-sporing rods is closely related to Brachythrix. Listeria monocytogenes is known to cause listeriosis and common on dairy products. Pantoia. This genus consists of gram-negative, non-capsulated, non-sporing striped rods, most of which are motile by Peritrichus flagella. They are widely distributed and are found on plants and in seeds, in soil, water and human specimens. Proteus. These enteric gram-negative rods are aerobes that often display pleomorphism, hence the generic name. They are typically of enteric bacteria in being present in the intestinal tract of humans and animals. They may be isolated from variety of vegetables and meat products, especially those that undergo spoilage at temperatures in the mesophilic range. Pseudomonas. These are typically soil and water bacteria and they are widely distributed among fresh fruits, especially vegetables, meats, poultry and seafood products. Although once the largest genus of foodborne bacteria, the genus has been delimited by the transfer of many former species. Salmonella. All members of this genus of gram-negative enteric bacteria are considered to be human pathogens. Serratia. These gram-negative rats that belong to the family Enterobacteriaceae are aerobic and proteolytic, and they generally produce red pigments on culture media and in certain foods, although non-pigmented strains are common. Serratia liquefaciens is the most prevalent of the foodborne species. It causes spoilage of refrigerated vegetables and meat products. Staphylococcus. These gram-positive, catalase-positive cocci include Staphylococcus aureus, which causes several diseases syndromes in humans, including foodborne gastroenteritis. Stenotrophomonas. These gram-negative rats are common inhabitants of plants and they have been recovered from soil, water and milk. Vibrio. These gram-negative straight or curved rats are members of the family Vibrionaceae. Several former species have been transferred to the genus Listonella. Several species cause gastroenteritis and other human illness. Yersinia. This genus includes the agent of human plague, Yersinia pestis, and at least one species that causes foodborne gastroenteritis. Common genera of foodborne molds. Molds are filamentous fungi that grow in the form of a tangled mass that spreads rapidly and may cover several inches of area in 2 to 3 days. The total of the mass or any large portion of it is referred to as mycelium. Mycelium is composed of branches or filaments referred to as hyphae. Those of greatest importance in foods multiply by ascospores, zygospores or conidia. Some of the common fungi are as follows: Alternaria, septate mycelia with conidiophores and large brown conidia are produced. The conidia have both cross and longitudinal septa and are variously shaped. They cause brown to black rots of stone fruits, apples and figs. Stem-end rot and block rot of citrus fruits are also caused by the species or strains of this genus. This is a field fungus that grows on wheat. Additionally, it is found on red meats. Some species produce mycotoxins. Aspergillus. Chains of conidia are produced where Cleistothecia with ascospores are developed, the perfect stage of those found in foods. The aspergilli appear yellow to green to black on a large number of foods. Black rot of peaches, citrus fruits and figs is one of the fruit spoilage conditions produced. They are found on country cured hams and on bacon. Some species cause spoilage of oils such as palm, peanut and corn. Aspergillus oryzae and Aspergillus zoae are involved in the shogo fermentation and the former in kozi. Aspergillus glucus produces a fermented fish product. The Aspergillus glucus, a Aspergillus restricus group, contains storage fungi that invade seeds, soybean and common beans. Several species produce aflatoxins and other produce ochratoxin A and sterigmatocystine. Botrytis, long, slender and often pigmented conidiophores are produced. Mycelium is septate. Conidia are borne on apical cells and are grey in color, although black, irregular sclerotia are sometimes produced. Botrytis cinerea is the most common in foods. They are notable as the cause of grey mold rot of apples, pears, raspberries, strawberries, grapes, blueberries, citrus and some stone fruits. Fusarium. Extensive mycelium is produced that is a cottony with tinges of pink, red, purple or brown. Septate fusiform to sickle shaped conidia that are called as macroconidia are produced. They cause brown rot of citrus fruits and pineapples and soft rot of the fig. As field fungi, some grow on barley and wheat grains. Some species produce geraniolin, phemonisins and trichothecins. Geotrichum. It is variously referred to as dairy mold because it imparts flavor and aroma to many types of cheeses, and as machinery mold because it builds up food contact equipment in food processing plants, especially tomato canning plants. They cause sour rot of citrus fruits and peaches and the spoilage of dairy cream. They are widespread and have been found on meats and many vegetables. Some participate in the fermentation of gari. Mucor. It is found in fermented foods, bacon and many vegetables. One species ferments soybean whey curd. Penicillium. It has been involved in the spoilage of fruit juice concentrates. It produces heat resistant spores. Typically, colors on foods are blue to blue green. Blue and blue green mold rots of citrus fruits and blue mold rot on apples, grapes, pears and stone fruits are caused by some species. One species, Penicillium roqueforti, produces blue cheese. Some species produce citrinine, yellow rice toxin, ochratoxin A, rubrotoxin B and other mycotoxins. Rhizopus, non-septate hyphae are produced that gives rise to stolons and rhizoids. Sporangiosperms typically develop in clusters, form ends of stolon at the point of origin of rhizoids. Rhizopus stolonifer is by far the most common species in foods. Sometimes referred to as bread molds, they produce watery soft rot of apples, pears, stone fruits, grapes, figs and others. Some cause black spot of beef and frozen mutton. They may be found on bacon and other processed meats. Some produce pectinases, and Rhizopus oligosporus is important in the production of oncom, bangruk and tempeh, which are fermented foods. Trichothecium, septate hyphae that bear long, slender and simple conidiophores are produced. Trichothecium roseum is the only species and it is a pink in color and causes pink rot on fruits. It also causes soft rot of cucurbits and is common on barley, wheat, corn and pecans. And some produce mycotoxins. Let us discuss now common genera of foodborne yeast. Yeast may be viewed as being unicellular fungi in contrast to the molds, which are multicellular. However, this is not a precise definition, as many what are commonly regarded as yeast actually produce mycelia to varying degrees. Some of the common yeasts are as follows. Brettanomyces. These ascosporogenous yeast shows terminal budding and produce acetic acid from glucose only under aerobic conditions. They cause spoilage of beer, wine, soft drinks and pickles, and some are involved in after fermentation of some beers and ales. Candida. Members of this genus are most common yeast in fresh ground beef and poultry, and Candida tropicalis is the most prevalent in foods in general. Some members are involved in the fermentation of cocoa beans as a component of kefir grains and in many other products including beers, ales and fruit juices. Dibaromyces. These ascosporogenous yeasts sometimes produce a pseudomycelium and reproduce by multilateral budding. They are one of the two most prevalent yeast genera in dairy products. Cluaromyces. These ascospore forming yeast reproduce by multilateral budding and the spores are spherical. It is one of the two most prevalent yeasts in the dairy products. Chloromyces species produce beta-galactosidase and are vigorous fermenters of sugars, including lactose. Chloromyces species contains coenzyme Q and is involved in the fermentation of coumaces. It is also used for lactase production from whey and as the organism of choice for producing yeast cells from whey. They are found on a wide variety of fruits and also cause cheese spoilage. Pichia. This is the largest genus of true yeast. Pichia species typically form films on liquid media and are known to be important in producing indigenous foods in various parts of the world. Some have been found on fresh fish and shrimp, and they are known to grow in olive brines and to cause spoilage of pickles. Rhodotorula. These yeasts are anamorphs of Basidiomycetes. They produce pink to red pigments and most are orange or salmon pink in color. The genus contains many cytotrophic species that are found on fresh poultry, shrimp, fish and beef. Some grow on surface of butter. Saccharomyces. These Ascosporogenous yeast multiply by budding and produce spherical spores in assae. All bakers, brewers, wine and champagne yeast are Saccharomyces cerevisiae. They are found in kefir grains and can be isolated from a wide range of foods such as dry cured salami and numerous fruits. Let us discuss now common viruses on foods. Viruses account for most food poisoning cases where a specific contaminant is found. Noroviruses, which are commonly called as Norwalk-like viruses or caliciviruses, are a group of viruses that cause a mild illness. It is the most common viral cause of adult food poisoning and is transmitted often from water, shellfish and vegetables contaminated by feces as well as from person to person. Rotavirus. It is the most common cause of food poisoning in infants and children and is transmitted from person to person by fecal contamination of food and shared play areas. Hepatitis A. Hepatitis A virus causes moderate illness with sudden onset of fever, loss of appetite, abdominal pain and feeling of tiredness followed by jaundice, which is a yellowing of the eyes and skin. It is transmitted from person to person by fecal contamination of food. In conclusion, except for a few sterile foods, all foods harbor one or more types of microorganisms. Some of them have desirable roles in food, such as in the production of naturally fermented food, whereas others cause food spoilage and foodborne diseases. Thank you.